And now, we move on to the scientific news of the day. Some researchers from the University of Vigo and Santiago de Compostela in Spain have been investigating how the effect of soil death and the attack of a well-known herbivore can affect the survival of an invasive plant. They titled this work the stress test, how soil death and aphid herbivory shape 𝘾𝐾𝐧𝙧𝙤𝙤𝐬𝑐 physiological responses. Let's connect to Noa, the principal researcher of this experiment. Hello everyone! Thank you for inviting me to explain this work. To make it more fluid and dynamic, I'm going to play the video I made and then I'll connect again. Let's get started! Invasive plants of the genus 𝘾𝐾𝐧𝙧𝙤𝙤𝙪 have a high phenotypic plasticity that allows them to live in many coastal environments, such as dunes, littoral forests, and cliffs, and to support biotic stress from herbivore pressure. For example, in Galicia, Spain, invasive 𝘾𝐾𝐧𝙧𝙤𝙤𝙪 species can be found in both deep and shallow soils, where native generalist herbivores like aphids feed on them. However, the mechanisms that allow 𝘾𝐾𝐧𝙧𝙧𝙤𝙬 to grow in shallow soil, even when they are under biotic stress, are unknown.
Hola, ¿podría hacerte una pregunta?
Claro, dime.
¿Te suena esta planta?
Sí, es una que crece en las zonas costeras de Galicia, ¿verdad? Y es que además es súper bonita.
¿Sabías que es invasora?
No, la verdad es que no tenía ni idea. Pero ahora que me doy cuenta, sí que es verdad que me tengo fijado que donde crece esta planta no suelen crecer otro tipo de plantas y cada vez crece en más sitios.
Pues sí, es invasora y es del género 𝘾𝐾𝐽𝑧𝙧𝙤𝙤𝙀. A mucha gente le parece bonita, por sus grandes flores amarillas o rosas, y la planta en sus jardines. Además es súper fácil de mantener. No le haces caso y no muere. ¿La has visto en algún jardín?
En los paseos de San Mil de Vigo hay muchísimas de estas plantas, sí.
Ya, pues muchas gracias.
De nada.
My cat also loves this plant. I don't know if has anything to do with the fact that it is called uña de gato in Spanish.
Hola, ¿te suena este bicho?
Sí, me suena ese bicho mucho porque me come todas las legumbres y vegetales. Me cago en un bicho que malo es.
Pues sí. Se alimenta del floema de muchas plantas, es generalista.
¿Y cómo se llama?
Esta especie se llama Aphis fabae y es nativa de Europa y Asia. Muchas gracias.
Gracias a ti.
As this lady said, aphids feed on different plants and kill them. It's considered a pest, so it could effectively attack 𝘾𝐾𝐧𝙧𝙧𝙤𝙬𝙀.
In addition, other experiments I'm conducting have shown that both species interact with each other, and 𝘾𝐾𝐽𝑧𝙧𝙧𝙤𝙬𝙀 can be found in deep soils, like dunes, as well as in shallow soils, like cliffs. To investigate how soil depth and herbivory affect 𝘾𝐾𝐽𝑧𝙧𝙧𝙀, we collected 60 plant fragments in Bayona, Vigo. We planted 30 in pots simulating deep soils. The pots were filled with sand, and 30 in pots simulating shallow soils. The pots were with a plastic barrier and 2 cm of sand over it, thus reducing the depth by 70%. After the acclimatization period, plants were subjected to different treatments. For each depth, we used as controls 10 plants. We treated with aphids other 10 plants, and we punctured with needle to simulate herbivory the last 10 plants. From time to time, an aphid was added to each plant in its treatment, and the plants in the simulated herbivory were punctured. In other words, if a plant ended up with 10 aphids, the plants in the simulated herbivory treatment ended up with 10 punctures. After 6 weeks, we measured morphological parameters like shoot and root biomass, photosynthetic parameters like Phyt2 and Phyno, and biochemical parameters like pigments and enzymes. Let's see the results. Shallow soils affected the aerial part more than the root part. For example, here you can see how it affected both length and dry weight. However, if you look at the specific root length, you can see that plants with less rooting space were more thickened or had more secondary roots, since per centimeter of root, plants had a higher root weight. To understand photosynthetic and biochemical parameters, I will first explain how stress affects plants. When plants aren't in optimal conditions, a war occurs in their cells called oxidative stress. And this war occurs because reactive oxygen species, or ROS, are generated. ROS would be like a harmful army that destroys the DNA. Antioxidant enzymes would be the army that defends the DNA. When the ROS arrive, the antioxidants will reduce the amount of them. But if the stress is continuous and high, eventually they can reach the DNA and destroy it, seriously affecting the photosynthesis and the general health of the plant. Photosynthesis is the process by which plants transform solar energy into chemical energy. It takes place in the chloroplast of plant cells, especially in the photosystems that contain pigments like chlorophyll. When light is captured by photosynthetic pigments, reactive oxygen species are naturally produced. In moderate amounts, these molecules are not a problem for plants, as they help to activate defense pathways. But in excess, like due to a strong stress, can be detrimental. In this study, we can see that control plants show high photosynthetic efficiency, low energy loss, and minimal activation of protective mechanisms, indicating healthy and unstressed conditions. Puncture plants exhibited the strongest signs of stress, with a pronounced decrease in 𝘊2 and a marked increase in 𝘊NO and 𝘊NBQ. This suggests that mechanical damage causes both photoinhibition and activation of energy dissipation mechanisms. Plants with aphid fabae also show stress responses, but less than puncture plants. Shallow soil tended to amplify negative effects of both treatments. As I said before, photosystems contain pigments such as chlorophyll and carotenoids. The stress the plants are facing is not reflected in each pigment individually. However, a high chlorophyll-carotenoid ratio indicates low stress, which is what you can see in the control plants, while those subjected to aphids and those in shallow soils were more stressed. ROS produced in the photosynthetic process must be neutralized, and that's the function of antioxidant enzymes. In particular, superoxide dismutase or SOD, or catalase or CAT activity, is key to mitigate the damage caused by them in chloroplasts. In this study, we can see that in control plants, antioxidant enzyme activity was low, in line with the absence of oxidative stress. SOD activity increased in puncture plants, suggesting an active antioxidant response. CAT activity also showed some high values. In plants with aphid fabae, SOD activity increased, and CAT also showed some peaks. Carpobrotus plants reacted in a different way depending on the type of stress. Plants puncture simulating herbivory showed the strongest stress response, while aphid attack caused less stress, in both cases suggesting activation of defenses rather than damage. Soil depth also matters. In shallow soils, plants develop less aerial part, activate less defenses, and suffer more. So, these results suggest that Carpobrotus has a complex and efficient tolerance or compensation mechanisms when it's under biotic stress, herbivory, abiotic stress, soil depth, or a combination of both stressors. All plants, despite stress and different adverse situations, grew well and, of course, survived. Wow, Carpobrotus is like a monster! Thank you very much for showing us your work. It's very interesting. Thank you for allowing me to show it. Soon it will be published, in case anyone is interested in knowing about other results not shown here. But I think the main ideas are the ones I show. So, thank you. Lovely! We'll be looking forward to reading it. Thank you very much, and see you soon. We say goodbye to all of you and see you in the next edition. Bye!